everyone, my name is Lily Vu. I'm a doctoral student in the Systems and Cellular Neuroscience PhD program. Today I'll be presenting a paper by Kim and collaborators at the Korea Institute of Science and Technology. The title of the paper is Transspinal Focus Ultrasound Stimulation Selectively Modulates Descending Motor Pathway. The descending motor pathway investigated in the study is the corticospinal tract. This neurological pathway is responsible for sending motor-related information from the motor cortex down to the spinal cord. Malfunctions in this pathway are associated with neurological movement disorders such as Parkinson's disease, which causes uncontrolled movements, often including tremors. The therapeutic approach investigated in this paper is transspinal focus ultrasound, applying ultrasound waves to the spinal cord. Advantages of this stimulation over techniques such as electrical stimulation and magnetic stimulation are high spatial precision and that it is non-invasive. There are three main research questions addressed in this study. Number one, how does focused ultrasound neuromodulation on the spinal cord affect the descending pathways of the corticospinal tract? Number two, how does the same ultrasound protocol affect the ascending pathway of the corticospinal tract? And finally, number three, could ultrasound neuromodulation be used to treat motor deficits? To address the first research question, microelectrodes were surgically placed into the area of the motor cortex responsible for hind limb movement, and stainless steel wires were inserted into the hind limb to record muscle activity. Areas T12 and L3 of the spinal cord were targeted in this experiment. The objective is to record muscle activity of the hind limb while electrically stimulating the motor cortex and varying the acoustic pressure and target of sonication on the spinal cord. This way, experimenters can observe changes in signal from cortex to hind limb in order to see whether or not sonication affects the descending pathway of the corticospinal tract. To address how the same sonication protocol would affect the ascending pathway, the role of the electrodes already placed in the mice were switched. The electrode in the hind limb was used for electrical stimulation instead of recording, and a stainless steel screw replaced the electrode in the brain for recording instead of stimulation. Thus, we deliver sensory electrical stimulation to the hind limb and record somatosensory evoked potentials from the brain. To address the last research question, an essential tremor was induced in mice using the Harmaline IP injection. The same recording and simulation setup used to investigate the descending pathways was used, and sonication on area T12 of the spinal cord was tested. As for the ultrasound applied, the focused ultrasound transducer was aligned with the spinal segment of interest and placed directly on top of the skin of the mouse. The experiments consisted of three segments: baseline, sonication, and recovery. During the sonication segment, it was delivered from the transducer in pulse trains lasting 200 milliseconds each. Electrical stimulation was then applied to either the motor cortex or the hind limb in all three segments. What Kim and authors found in this study is that with sonication of area T12 of the spinal cord, motor signals from the cortex to the hind limb were inhibited. This effect was transient. The signals returned to baseline levels in recovery. You can see this in figure 2b, where the EMG response of the hind limb decreases with increasing acoustic pressure. At 0.8 megapascals of acoustic pressure, EMG signals are close to baseline. At 1.4 megapascals, EMG signals are slightly reduced, and at 2.2 megapascals of acoustic pressure, EMG signals are almost entirely reduced during sonication. With sonication of L3, motor signals from the cortex to the hind limb were facilitated. This effect was also transient. EMG hind limb response increased during sonication. When applying sonication to either T12 or L3 and recording from the motor cortex while stimulating the hind limb, there is no significant change in somatosensory evoked potentials, showing that the effect of ultrasound neuromodulation on the spinal cord is directional. It affected the descending pathway, but it had no apparent effect on the ascending pathway. For the last objective, Kim and authors found that application of spinal ultrasound stimulation on essential tremor mouse models reduced their motor deficit. EMG recordings from the hind limb show decreased amplitude during sonication. Take-home messages are that transspinal focus ultrasound stimulation could be a possible clinical treatment of motor deficits that is both non-invasive and spatially precise. The effect of spinal ultrasound stimulation is pathway-specific, location-dependent, and correlates with acoustic pressure. Sonication affected the descending pathway, but not the ascending pathway. Applying sonication to T12 inhibited motor signals, while applying sonication to L3 facilitated motor signals. And increasing acoustic pressure increased both facilitation and inhibition effects. For spinal ultrasound stimulation to become clinically useful, future studies should investigate how to maintain a lasting effect from sonication and how to only inhibit unwanted movement. Thank you everyone for watching my presentation of this paper describing how transspinal focus ultrasound stimulation can affect descending pathways on the corticospinal tract.